We're ready to go. So good evening, everyone. I want to welcome tonight Dr. Lisa Boyette from Recursion Pharmaceuticals. She is the Senior Director of Medical Affairs and is right now the main person that we talk to about their very exciting REC-994 program, which has been moving along and is coming soon to a next stage. I am not going to talk any longer. I'm sorry. One more thing. If you have questions, please use the Q&A rather than the chat to put them in, because that's where I'm going to be looking for them toward the end. So please feel free to ask questions, but please use the Q&A to do so. Please.
Well, thank you, Connie, for inviting us to come and talk with all of you about the REC-994 program that we've been working on. I'll be very interested to hear what kind of questions people have and do my best to answer them. I'm going to share some slides so that I can tell you a little more about Recursion, about clinical research, and about how we're designing this REC-994 program to answer some questions about CCM. So we have named our study of REC-994 in CCM the Sycamore study. So that's something you can look for in the future, the name of the clinical trial. As Connie mentioned, I'm Lisa Boyette, and as the head of medical affairs, my job is really about building and maintaining our relationships with external stakeholders like physicians who participate in our clinical trials, patients who participate in our clinical trials, advocacy groups. But I also happen to be the project team leader for our REC-994 CCM program. So I am really dug into the science and have a real fondness for this particular program. The first thing I'd like to spend a little bit of time on is how Recursion Pharmaceuticals is different from maybe some other pharmaceutical or biotech companies out there. For those of you who've heard this before, I will not belabor it. It won't take too long, but it's really pretty interesting where we are as a group at this point in time. So the philosophy of our company is that technology has been disrupting all of these other industries, and drug development is an area where we could probably use a little more disruption. If we look at the cost of drug development over the last couple decades, we don't really see it coming down. And I apologize, one of my lines is just not plotting here. There's a red line that should be up here showing you that the cost is going up per study rather than coming down. So what can we do to put ourselves on a new trajectory? There it is. And the reason that we as an industry have had trouble doing this is because biology is really complex. So if you think about the kind of other industries where we can rapidly scale or we can disrupt paradigms by increased application of technology, biology, it's a little hard to figure out exactly how we get started with that. And this picture that's zooming out in the background is just one kind of functional collection of enzymes that works within cells. So if you imagine this times every cell type in your body, and then how those things are sometimes disrupted in disease states, it's just an enormous quantity of information we have to understand to make better therapies. However, we have gotten started with building a system that is aimed at doing exactly that, understanding that enormous complex quantity of information that drives human health and in places where it gets disrupted human disease. So we have a lot of really sophisticated tools. Most of you have probably heard of CRISPR. We use tools like CRISPR as well as CRISPR to make changes in experiments we do in little 2D cell cultures in a dish. We have built massive automation capabilities. So we have lots of robots that moves those dishes around in the lab, moves them over to the microscope for pictures to be taken. And then the other things that have enabled us to really scale our approach to taking in all this complex biological information are, some of the developments over in the tech industry, like cloud storage, compute power. And then our system is now in a mode where we're using artificial intelligence not only to analyze the results that we get from the individual experiments that we do, but also to look across all of the experiments that we do and start telling us where biological relationships are that maybe we haven't hypothesized about yet. The result of having built all of those tools is that we do a lot of experiments now. So we have done over 82 million experiments to date. That's about 1.5 million experiments a week. If you want to picture how much data that generates, taking pictures of each of those experiments, you can imagine this 9 petabytes of data as all of the movies that have ever been made in human history in high definition. So feature-length films to put together. We are working constantly to onboard new types of human cells into our system. So we got started with one type of cell, and then we onboarded another type of cell. And what that does is allow us to really ask the question about whether the genetic changes we're putting into the cells to make our models are relevant to a particular disease. We want those models to be as relevant as they can be. And then from all of this information, we've now managed to capture what we believe are 179 billion distinct relationships between different human genes, chemical compounds. Those are, potential drug candidates that we've screened using a set of maps that we're building of biology. Let's see if this will advance. Just to kind of give you a feel for what this looks like in the real world, here's some pictures from our lab. So we have these big, beautiful white laboratories inside our headquarters in Salt Lake City with all these robotic stations and little arms moving plates around. And the people are really there to just supervise at this point. It's pretty amazing. And then all the way over to the right, where you're seeing this plate populate with different little flashes. This is an example of, those little experiments that are done, and we might change one condition in each of those wells. And then we look at what it does to the cells that are growing in that dish, that we have perturbed a particular gene or that we've added a particular chemical compound. To give you a look at kind of the tech side, how we analyze all of this data, we have a top 100 supercomputer at Recursion, and that's top 100 in the entire world. So if you think about the kinds of organizations that keep supercomputers lying around, that's pretty exciting to be a part of something like that. And from all of the analysis, all the compute power that we have in that supercomputer, we can generate, some pretty interesting data about relationships between genes and chemical compounds when we haven't necessarily done the direct comparison in a real experiment. So now we're doing experiments in silico. And that gives us some insight into exactly the right experiments that we should spend our time and our energy and our money on doing. And the result of all of that is that we fail compounds for a particular disease state earlier, which means we spend less time and resources and money on things that aren't going to work. And we can more quickly narrow down finding a number of drug candidates that have a better chance of working. So we can do this more cheaply as a result of all the work we're doing in silico now. And the most important thing for us as a company, and the thing that I think matters most to all of you, is that this kind of approach enables us to crank down on how long it takes to get through some of these stages of drug discovery. The result in terms of programs, I just wanted to give you a high-level snapshot. CCM is one of the rare diseases that we currently have in active clinical development. It is the program that launched the idea of Recursion. The birth of our CCM program is really the story of the birth of Recursion as a company. But alongside CCM, everything else that we have in active clinical development right now is also a rare disease. So as a company, we have a real commitment to rare disease. And we have worked to really build a team with expertise in how to do rare disease trials and serve rare disease research communities, which includes all of you. I threw this in just so you can kind of see where we as a company sit alongside everybody else who's doing tech-enabled drug discovery right now. Recursion is leading the pack And I thought you would be excited to be, a part of a discussion with a company that is really out over their skis, trying to push the envelope as much as possible on behalf of patients. We have grown tremendously over the last year. We are now at 300 plus employees, and we're a company that's committed to diversity. So we make sure we have gender and ethnic representation in all functions within Recursion. Huge priority for us. I'd like to take a minute and just touch on a few key points about clinical research. Before I do that, Connie, are there any pressing questions that we should address before we move on?
Only one person has asked a question, and they have asked two-word question: quantum computers.
So these are not quantum computers. It is a very large set of more traditional servers, but we have people whose entire function at Recursion is to make those computers run more efficiently. Definitely not my department. I can't speak to the particular hardware that we have in-house, but we're not using quantum computing yet. Great question. So let's take a couple minutes and talk about just clinical research as a general concept, because I think that will set the stage well for talking about what we've done so far in our CCM program and what we're planning to do next. This is a diagram that I pulled from FDA. So this is methodology that would apply to any kind of drug development process. A company comes up with a candidate. When you see the word sponsor, that's the drug company that they think could be approved as a drug in the United States. They do a lot of work in animals to make sure that that drug candidate is going to be safe, and to try to get some idea of whether it is relevant to the disease that they're investigating. If all of that lines up and looks good, they submit to the FDA what's called an investigational new drug application. Following review of that, the regulatory agency in our country will give that company permission to start doing work in humans. And I just want to highlight this process because I think there's a lot of misunderstanding about why it takes so long to develop drugs. And a lot of us in industry love to just blame regulators for things being slow, but really regulators are here to keep us all on the straight and narrow and make sure that we don't do anything in people that's not ethical and safe. So once FDA decides that they feel good about what's been submitted with the IND, phase 1 studies will begin. I'll come back and talk about this for REC-994 specifically in a minute. Usually these are smaller numbers of either healthy volunteers or, depending on the type of drug, sometimes patients. And it's just to get an idea of how the drug acts once it's in the body, what the body is doing to the drug, what the drug is doing to the body. And then once that is done and the company has a better understanding of how their drug is working and that it's not causing any major safety issues, we move on to what's known as phase 2 studies. This is usually where you're working in patients for sure, and you really start asking questions about what dose of a drug is going to be relevant for making the disease state look better potentially, what dose of a drug is going to be tolerable, meaning it doesn't cause people to feel ill or have a whole bunch of side effects. And these are larger numbers of patients. One exception to that would be in rare diseases where maybe we can't put hundreds of people into a phase 2 study. So then sometimes we will see smaller numbers of patients. If at the end of phase 2, a dose or set of doses has been found, at which the data all suggests that maybe this drug would be helpful in that disease state, then you move on to what are called phase 3 studies. And this is where the rubber hits the road. This is where the big money gets spent, lots of people, longer periods of following those people on drug and really trying to understand if taking that pill is going to impact that patient's disease state for the better. And if those findings can be generalized to some larger slice of the patient population or an entire patient population. So that's the drug development process in the United States. Following all of that, there are some negotiations about exactly who's going to be able to get a prescription for this medicine, which is about the label being written. And then there's all kinds of quality to make sure we manufacture drugs in a certain way in this country. And then if a drug is approved, and it starts being prescribed to people in the clinic, then we enter into what is known as a phase 4 trial, where the company continues to collect data about how people are doing on a drug, so that if something starts going wrong further down the line, we capture that and we can act accordingly. So having said that, I want to show you our brand new clinical development team at Recursion that has been built out over the last year. And this is not the entire team. This is the entire team that's working on this program specifically. But as you can see, it's a lot of people and they all have different sets of expertise. You know, I'm an MD PhD scientist and I lead this particular team, but we have a vice president of regulatory who's very experienced. We have some experienced clinical people, operations people who actually make our trial real on the ground and make sure that all the interactions with the hospitals and the doctors and the patients go the way they're supposed to. This is our boss, Ramona Doyle, who's our chief medical officer and is taking care of rare disease patients her entire career as a transplant pulmonologist, and now leads our team developing drugs for a whole bunch of rare diseases. So it's a dynamic, really smart, exciting group of people to work with, and all at Recursion because they want to be in a place where there's a commitment to great science and a commitment to serving patients. So I just wanted you to see kind of the breadth of expertise that goes into a program like this. All right, let's talk about our trial specifically, because I know that's what everyone is here to learn more about. I don't need to tell all of you about the disease state. I just wanted to mention that we have really chosen to focus in not only on understanding what REC-994 might do to the disease state in terms of, you know, how we can look at it on imaging like MRI, but we also really want to understand how REC-994 might impact symptoms that patients are living with. You know, and it's through our relationship with Angioma Alliance and the work that has been done to develop a CCM specific patient reported outcome instrument, which you're going to hear a little more about later. You know, that's a particular survey that has been made just for this disease to really capture what kind of journey people are having with their disease, or if they go on therapy. That's important. We understand that bleeds are not as frequent in this disease state as they are in some other types of neurovascular diseases, and that it's important to patients to have improvement in their symptoms. So we're looking at all of those different facets. And, you know, our ability to design a great study has been really dependent on talking to people who actually live with the disease. So thanks for all of the input that you've given over the last several years. It's made a big difference in our study design. I want to show you what kind of evidence we generated along the way to convince us that REC-994 might have a role to play in CCM. Some of you who've been attending these talks for a while may have seen some of this before, but for those of you who have not, we did a number of experiments in animals after the initial finding that REC-994 might work for CCM. And we looked at vasodilation, which is one of the features of vascular permeability. We looked at a different permeability model with genetic mutations that cause CCM-like lesions in that animal. And then we also looked at the number of CCM lesions in models with the same genetic mutation as we see in the human disease. And in all of these experiments, what we see, and over here, you're looking at the difference between these plotted lines. REC-994 is the black dots. So you can see, REC-994 is the purple. They go across like this, because it should be up here with wild type animals. So when these animals got the drug, they were restored back to what a normal animal looks like. Same thing here. We had a disease state, and then when we gave the animals the drug in white, it was restored back to about a normal level. And then in terms of number of lesions, again, as we increase, or we add REC-994 into these animals, which are the white bars, at every different bracket of lesion sizes, you can see that there are fewer lesions in the mice who got REC-994 than in the mice who did not. So seeing all of that was part of our decision to move forward into a program with humans. And this has all been published in journal articles. So, you know, all the scientists in the community that work on this disease have taken a look at this data. This is showing you the same thing I showed you on the last slide about the reduced number of lesions in the animals, but here I'm showing it for both CCM1 and CCM2. So given all of that data, we started planning for a study that would target a patient population with genetic mutations in the CCM1, 2, or 3 genes. And we plan to enroll our first patient into that study, which will have all the bells and whistles. So it will be blinded, meaning that the doctor doesn't know whether the patient is getting which dose of the drug or placebo. It will have a small placebo arm to make sure that we're really understanding the difference between getting this drug and not having it. And this will be, if things continue the way that we think they're going to, this will be the first industry-sponsored trial for symptomatic CCM ever, which we're really excited to be a part of. This is a drug that we don't have a lot of safety concerns about. It has been tested in a phase 1 study, and I'll show you some data from that in a second, and it was well tolerated. People did not have a lot of side effects. So here's a summary of the findings from the phase 1 studies that were done. So there was a phase 1A study done in 2019, and this was done in healthy volunteers. And then there was another phase 1 study done in 2020, also in healthy volunteers. And really the idea with these studies is to give gradually increasing amounts of the drug, first as single doses, and then as multiple doses, like you take the drug every day in a row, and make sure that it was safe. And really understand if there was any upper limit where people start to have side effects from the drug. And what we found is that the drug behaved in the way that we expected it to. So when I say there were dose proportional pharmacokinetics, it means when we gave more of the drug, we saw more of it show up in the bloodstream. And that was exactly the way we expected that to happen. It was well tolerated up to some pretty high doses, which is great. And then all of the adverse events, so these are safety findings or side effects, were mild or moderate, and most of them were determined to not even be related to being on the study drug. So because we saw this very consistent, reassuring data in the phase 1, we decided to move forward with a program that would bring this molecule into a study with CCM patients. And this is kind of just summarizing what I said on the last slide, so I'm going to skip over this. So here's what the design looks like for our phase 2 clinical trial in CCM. People entering this study have to be over 18. They have to have CCM lesions that can be seen on an MRI, and their physician has to feel like they are symptomatic, meaning you have a lesion and you're having some kind of symptoms like headaches or seizures, and your doctor feels like those symptoms can be attributed to your CCM lesion. For people who have multiple lesions, same kind of idea. That's a determination that's made by the doctor putting that person in the study. And then those people will get randomized into taking a low dose of REC-994, a higher dose of REC-994, or a placebo. Because we have a 1-to-1-to-1 randomization into these arms, meaning if I'm a patient coming into the study, you know, I kind of get a number, and depending on it's a 1 or a 2 or a 3, that's the arm I will end up in. Because we have 2 treatment arms and only 1 placebo arm, any patient coming into the study has a 2 in 3 chance of ending up on study drug, active study drug. So we treat everybody for a year, and then we do a follow-up period where we continue to collect some information about how everyone is doing, how they're feeling, how their disease state is progressing or not progressing. So that's the general design for the study. Unfortunately, I can't share a lot of details beyond that tonight, but we do expect to have more information very soon. So I'm going to stop there and make sure that we have plenty of time left for questions, and it looks like there are a couple.
Yes, thank you. And I'm sure more will be rolling in. One person has the same question that I have, because there was some contradiction in your slides. Is the trial only for people with the hereditary form of the illness, or can people with a sporadic form of the illness also participate?
Oh, that's a great question. So people with the sporadic form of the illness can also participate. And as part of the study, we'll do some testing to understand, if they have a hereditary form of the illness that has been missed. But yes, people with the sporadic form of the illness will also be allowed to participate. There are some other what we call inclusion and exclusion criteria. So sort of who's eligible for the study. So it's not a guarantee that anybody with any CCM lesion would be able to participate, but it's not restricted to people with hereditary lesions.
Okay, that's what I wanted to hear, because that's not what one of your slides said. One of your slides said that it is, so good. Next question was, if someone can't tolerate the high dose, if they get put on the high dose in phase 2, is there any option for them to move into a different leg, or do they just leave the trial at that point?
I think that's something that we'll have to answer questions about once we start enrolling people into the study, and we'll have investigators speaking about that at future meetings.
Okay. And then is this a double-blinded trial or a single-blinded? And the question specifically is, will a patient know if they're on placebo?
Right. This is technically a triple-blinded trial. Nobody ever uses that term, but what I mean when I say that, and what we usually mean when we say double-blind, is the patient doesn't know whether they're on active drug or on placebo. The doctor who enrolled them in the study doesn't know whether they're on active or placebo. And we as the sponsor don't know while the trial is ongoing.
Okay. And then you unblind at the end. And if somebody has dropped out because of side effects, you only find out then whether or not it might have been the placebo causing the side effects.
Correct. That's right. Mm-hmm.
Okay. And then someone else is asking about,
and Connie, if I could, I'd just like to explain why we do things that way. The reason we stay blinded is because we don't want to impact the quality of the data that we're capturing. We work in a number of rare diseases where, especially the doctors who take care of these patients, want them to get better. So, if they know that they're on drug, that can influence how they interpret how the patient's doing. And so we all stay blinded to what's really going on until the end. And then we look at all the data and try to understand as much as we can what the truth is.
Okay, and that makes sense to me. So next question is about imaging. Will you be imaging the study participants before and after and during?
There will be imaging. I can't say more specifically beyond that at this point what all there will be, but there will be imaging, yes.
Okay. Are there particular locations for lesion location that excludes them? And I think that goes along with someone else asking if someone has a single lesion, but it's in the spinal cord, are they eligible to participate?
That's another question that I will have to answer at another meeting with all of my doctor colleagues around me.
Okay. What is the target date for the completion of the first set of 52-week data?
Say that again. Sorry.
So someone wanted to know it's phrased a little bit interestingly, but they wanted to know, when is the first person going to be done with their phase 2? I guess, what is there a target date for that?
Right. There will be a target date published for that on clinicaltrials.gov when we register the study there, and that is coming very soon. And that is the earliest information. That's the earliest time that date will be made public. Okay. So I'm required to wait until that hits clinicaltrials.gov.
Okay. And how would a patient enroll?
Oh, so once the study is actually opening, we will come back to Angioma Alliance and we will say, here are the first few sites that will be opening, and here are the sites that we anticipate opening in the following months. It's our goal to make this a study that is widely accessible to people across the United States. So I can't share with you what the sites are right now because we're still negotiating contracts with all of them, but as soon as we can, Angioma Alliance will be one of our first stops to make that information available.
And you're not looking at like widely available doesn't mean like 4 sites. You're looking at a number that's north of 4 or 5.
Correct. Yes, it's a larger number of sites.
Okay. And how many people are you planning on enrolling?
I don't know if we have made that information public. I don't believe that we have. So it's probably not up to me tonight to make that information public, but as soon as the study is registered into clinicaltrials.gov, there will be a lot more information available.
Okay. And it's going to be, just so everybody knows, it's going to be first come, first serve. So, think about it once it opens. It might behoove you to get in sooner rather than later.
Yeah, and Connie's already thinking about it, and your doctors are already thinking about it if they're investigators that we've been talking with. So some of you may also be approached in clinic if you're, you happen to get your care at an institution that's going to be a site.
Yeah. Next question, what side effects could be expected?
Off the top of my head, the side effects that were observed were really mild. So some people and keep in mind, we have only used REC-994 in healthy volunteers. So there were some people that reported headache. In most cases, it was determined not to be related to being on study drug. We didn't have any side effects that emerged as consistent across that healthy volunteer population.
Okay. Is the study open to Canadians?
If they come to a US site to enroll, we don't have any restrictions on who participates at a given site.
Okay. And there probably will be sites close to the border. You probably don't have to go to Florida.
Right. Yeah.
Is this drug expected to reduce the size of current lesions?
We don't know. That is one of the key questions that we will ask as we continue to study it.
Are there any other drugs that a patient might be taking that could disqualify them from participating in the trial? For example, if they're already on statins, which are being tested already.
In general, and this is not specific to this particular study, in general, we don't allow people to take experimental medications for the illness that's being studied at the same time they're taking a study drug. So I can just say that as a generalization. Okay. And there will be a list of any medications that, people would have to come off of or would disqualify them from participating.
Yeah. So with a lot of trials, there's a washout period, right? You can have it, but you need to.
Yes, if someone is on something that would keep them from participating, we give them an opportunity to go off of that drug and let it completely come out of their system, and then they could become eligible.
Okay.
That doesn't apply to everything, but that's like a general principle.
Okay. I think this is a question that goes back to the size of the lesions, and it has to do with the mouse data that you showed. Based on the mouse data, the hypothesis is that the lesions would decrease in size, correct? I can explain.
Again, we don't know what to expect in humans. 1, mice are not humans. 2, and then the mice in that particular experiment that I showed you had been receiving this drug their entire lives. So it's hard to say, based on seeing data from mice that have received the drug as some of their lesions were forming, what that means for people taking the drug with lesions that are already formed. And that's why we've got to do the experiment in people who have the disease.
Okay. All right. Any idea about an expected start date?
I would love to tell you more. I cannot tell you more, but again, we will be back very soon with additional news.
Okay. Should we talk to our doctors about this now to see if they know about this?
I think it would be fine to put it on their radar, and what they're probably going to say is, no, we don't know about this yet, unless they happen to be one of the sites that we're talking with. But yeah, absolutely. Let your doctor know if you want to participate in clinical research, and start that conversation now, so that when the study is open, you guys are prepared and have already talked about the possibility of a referral or whatever you need to do.
And also, just because you're participating in a trial doesn't mean that you need to transfer your care to that place. You can keep your care if you're happy with it where you are, and just go to the trial site, right?
That is absolutely correct. And I would also say it's a good thing to talk with your own physician about whether they feel you would be a good fit for clinical research, if they would be comfortable with you participating in clinical research, because there can be a lot of different things to consider there, and your physician knows you.
Another question about taking other meds. If you are being treated for seizures and go in the study, do you need to stop taking the anti-seizure meds, or do you continue taking them?
Again, I'm not really at liberty to talk about all of the study specifics. However, for people who come into the study, it would be unethical for us to put them in a situation where they had uncontrolled seizures. So that will not be a scenario in this study.
Do you expect that there will be a site in the Northeast? Like, are you thinking that there's going to be regional sites?
Yes, we are planning for regional representation across the United States.
Okay. And
you will not have to all travel to Salt Lake City and meet us in person and go to, a local, or all go to Florida, or no.
Okay. All right. And then the last question oh, nope, they're coming up. Someone has asked this question again. So are there sites already known, and how would we know what facilities are participating?
There are no sites that have been formally opened yet. And if you want to know what sites are participating, that information will, at least the geographic area, will be posted on clinicaltrials.gov later. And we will also provide it to Angioma Alliance for distribution.
Okay. Someone says, damn shame it does not extend to Australia.
It's early days. Start somewhere.
Yeah, well, and it's phase 2, and with phase 3, there is, there will be different opportunities. I'm sure you can't say yet whether.
I can't say, because I don't know. We have to get through phase 2 and kind of see what we have. Does the data suggest that this is something helpful that should be continued in further studies? But when you do a phase 3 trial in a rare population, you have to go more places to get enough people to do a study that's meaningful. So I would expect, for most rare indications, expansion beyond where the phase 2 took place.
Okay. One other. Ooh.
And just another, kind of as another point of learning there, sometimes it's great in a phase 3 study, again, not specific to this program, just as a general principle, to go to different countries. Kind of standard of care can be different in different countries. And maybe you run a phase 2 trial in one geographic region and you get great results, and then you try to generalize that to the rest of the world and you get a little more of a mixed picture. So yeah, those are important things to learn in larger studies.
Someone wants to know, does this apply to individuals with who have multiple lesions but also have a DVA? I think the question is more about the DVA. Can you be in a trial if you have a developmental venous anomaly?
That is above my pay grade, and that would be up to the investigators who are actually enrolling patients into the study to look at the inclusion/exclusion criteria that we have provided and determine if any individual's set of disorders or symptoms makes them a good fit.
Okay.
And I say that, not just, I can't talk about all the specifics of what are in the protocol, but the people who are doing these evaluations are neurologists and neurosurgeons. I'm not a neurologist or a neurosurgeon, so that's kind of a specialist level decision.
Okay. And then a more general question, are there other diseases this drug could potentially help?
This drug has actually been used for a different disease. A topical form of this drug has been studied in preventing skin damage during radiation treatment. So the short answer is yes. And I think the longer answer is it remains to be seen whether we will identify additional diseases that we might put this drug into trials for. And it's a compound that other companies have studied. So it's possible that another company would put it into a trial for a different disease.
Someone else is asking this all about inclusion criteria tonight and exclusion. What about someone who had radiation to the brain?
I'm going to make all of you a deal. We're going to come back and we're going to do another one of these webinars after the study opens. We'll answer all of your questions. I'll bring along some experts, and we'll leave no stone unturned.
Okay. Then someone else asking when, when is that? And so again, we don't have an answer for that yet. I would like to share what people can do now, though, which is participate in the development of the CCM-Health Index, which is part of this trial as well. It's been a project that we've been working on for multiple years with the support of Recursion, and we will, Angioma Alliance and the University of Rochester have partnered on this. The University of Rochester is actually the place where it's being developed, but it's only being developed because you all are participating in the development. Starting probably next week, if we can just figure out one more answer about international participation, we will be announcing the next phase of the development. And that phase is going to require the participation of at least 400 patients. So we need all of you. And what this phase involves is taking a survey, part of which is the CCM-Health Index, but it also includes a different index too, 3 times, taking it once, and then 6 months later, responding to the email that you get to take it again. So you click the link and you spend the next 20 minutes of your life to 30 minutes of your life doing it again. And then at the 12-month point, again, one more time, going through the survey and doing it one last time. We're looking to see, we're looking at the change of answers and responses over time, how those compare to people's change and how they're feeling. We're looking too to compare this with another instrument that already has some data behind it. So it's really important that we get, excuse me, that we get participation. And I just today worked out what I think is the last of the criteria aside from that question on international participation. And right now it is, and international is about GDPR. So it's really just European that is the question. The participants must be 18 and they must have had a cavernous malformation at some point in their life. It's even for this stage, okay if they had it removed because they're tracking that. And it's okay if you only have one in your spine for this part of it. You need to be English speaking or able to read English so that you can respond to the questions and be willing to do this. So it's an relatively easy thing. It's over the course of a year. You'll probably give about 90 minutes of your time, but it is the most critical thing that you can do. Once we have this tool, it can be used in this trial, but it can be used in other things too. This would be a CCM specific tool that, so we wouldn't, what we've been doing since before this is just hacking together tools that are used for generic neuro diseases or for stroke or for, things that aren't exactly us. And this would be the first time that we would have a measure that where we would understand the patient experience that is specific to CCM. So I would encourage you, when you see me post next week, when you get the email in your email box from our patient registry, when you look at Facebook and see us posting there, please just click the link, take the 20 minutes, do the survey, and be a part of the solution.
Yeah, and I just want to emphasize how very important it is to have instruments like this that are tailored for a particular disease. We can have the greatest drug in the world, and if we have no way to measure whether it's making a difference, it's really hard to prove that the drug is working, and people should be allowed to take that drug. So critically important for advancing clinical research.
Yeah. And someone in the Q&A is asking about the link that I keep talking about. We'll make the link available next week as soon as we can know whether or not the Europeans can participate in this as well. I don't know it off the top of my head. It's something CCM-Health Index. And we'll put it out to our patient registry members. You'll get it in your email box. So join the patient registry, angiomaregistry.org. You'll be ahead of the game. A week from now, you'll get it in your email box, or you can keep monitoring Facebook to see when it's going to come there. Other questions? Oops. Could this be approved for children someday? That was another question.
Potentially.
And
We, in general, there are some exceptions to this, but in general, we try to see how something works and how safe it is in adults. And once we have that confidence, then if it applies to children, there's a path for pursuing expanding use of a particular medicine to children.
yeah, we certainly hope that we can take this medicine all the way through phase 3 and then into pediatrics. That would be awesome. So, okay. Someone else is asking when the recording is going to be online of this. We'll have it up probably in the next 2 days. I'm also hoping that Lindsay or Darla is here so that they can drop a link in the chat for the support group for people to register. Lindsay just did. So if you look in the chat, there is a link there. I am looking to see if there are other questions. And right now I'm not seeing any. If you have one, please put it in quickly. Well, I can just say it's a pleasure working with Recursion. This is a company that is very patient focused, and we've been watching their CEO, Dr. Chris Gibson, move this forward from the time that he was a graduate student at the University of Utah. And so it's been, it's just rewarding to continue to watch the commitment that Recursion has to the product. Hopefully will become a product, and to the patient community and to helping our people.
Yeah. And, Connie, we really feel the same way. We love working with Angioma Alliance. I'm sure most of you don't know, I actually meet with Connie in a good month, twice a month, on a regular basis, because we want to make sure that everything we're doing is in sync with what you all are planning and expecting, and vice versa. We're a diverse group of employees at Recursion. We have far fewer doctors than we have software engineers. And most of the people who joined Recursion wanted their work to mean something. These are people who could have gone to join an advertising firm or could have gone to one of the tech companies. And I hear over and over again, oh, I didn't want my work to just be about selling things or generating clickbait. I wanted to do something that was going to matter for people who are actively suffering or watching their family members suffer with a disease. I got into rare disease work because I have a sibling with a rare disease. So I used to work in a nonprofit organization very much like this one. We're a group of people who've all gravitated to this because we want to do something that matters for patients. And it's just a pleasure to work at a company where I hear people talk about service to patients on a daily to weekly basis. So that's a very real thing.
Yeah, thank you. One more question. How can we get genetic testing? That can happen a variety of different ways, but one way to do it is through us. If you register in our patient registry, and as part of that, one of the questions is, are you interested in genetic testing? And you answer that yes. I will personally review what your registry responses look like. And if you have multiple cavernous malformations that cannot be explained by the presence of a DVA or a history of brain radiation, then you are eligible to get free genetic testing through us. I would send you a form packet, and one of those forms needs to be signed off by the doctor who is going to receive the results of the genetic testing. You then get those forms back to me. I get them over to the lab with a test requisition, and they send you a saliva collection kit in the mail. And so in the privacy of your own home, you can spit into this tube and just mail it right back. And about 4 weeks later, the results come to me, and I send them to your doctor and let you know to contact your doctor's office. So that's one way to do it. But again, if you have to have multiple lesions that can't be explained by a DVA or a history of brain radiation. If you only have one lesion that's been seen so far, what you need to do is go the next time you see your neurologist, your neurosurgeon, whoever, make sure that they have included an SWI sequence in your MRI. That is the most sensitive of the sequences that they do. You all probably looked at your MRIs and seen the T1 and T2, et cetera. Well, the SWI is sensitive enough to pick up a second lesion if there is one there. And if there isn't one there, then 99.9% of the time, you do not have a genetic form of the illness and don't need genetic testing. It wouldn't make any sense because a negative result on genetic testing would have no meaning. We still have, even the people who have multiple lesions, about 5% of them don't get a result from our currently available commercial genetic testing because we're just not to that 100% level in technology yet. So negative result would mean nothing if you had only one lesion. I should be checking here. I think those are all the questions we have. Again, please everyone look in the chat and see if there are, check the link for the support group that's coming up and go ahead, click that and register for it. There's always good conversation happening. And if the group gets big, they can break out into several groups. We have one more question, and I'm not sure, Kristen, that I understand the question. It says recursion means the ongoing trials, and I'm not sure which ongoing trials anymore that you were talking about. I missed it when you typed it, but yeah, we want ongoing trials for for this. We want phase 2, phase 3, and then pediatric trials. So, any last thoughts?
Just that I would love to join you all for a few minutes if you feel like having a visitor, because it would be nice to meet you. I can't see anyone in this webinar format, and thank you for having us. It was our pleasure.
You're welcome. Just go ahead.
And the only other thought I'll leave you with, Connie, is just that on my side of things, it's my distinct privilege to represent that whole team of people who's been working for a long time to put this program together, who helped me put all of these slides together. And it's a team sport, and we're all pulling for all of you and for the study. Thank you.
All right, so Dr. Boyette, you need to click the link and get yourself registered. That's the only way into that support group. And in the meantime, I'm going to say goodnight to everyone and thank you so much for coming. If you have additional questions that come up, tomorrow morning, you think of something, just email me and I will try to get you an answer and get back to you. So thank you, everyone. I'm going to stop the recording.